Hi, my name is Allison Driscoll, and this is my marine biology final. I had questions 1 and 8 to answer. So the first question I had to answer was define what coral restoration is, and then discuss an example of one method being implemented in coral restoration. So coral restoration is the process of taking healthy fragments of coral and putting them back into the ocean, generally in areas where the reefs are suffering, in order to replenish the dwindling population of coral reefs. And the population of coral reefs is on the decline because an event called coral bleaching. And basically, coral bleaching occurs when the ocean's temperature rises at about 2 degrees Celsius. And this causes the corals to essentially become stressed out. And I'm not going to get too much into the details of it, but this basically causes the corals to get rid of their nutrient source and they end up dying eventually. And because corals are such an important part of our underwater ecosystem, they provide a home and structure for so many marine organisms. It's really important that we help replenish the coral reefs that are dying in order to sustain our underwater ecosystem. So, a few methods that are being implemented to do this to help restore corals is microfragmenting and the discovery of supercorals, which was the idea that the late Ruth Gates had. So, microfragmenting is a technique in which a large coral reef will be cut into really, really tiny, maybe 1 centimeter by 1 centimeter squares. And this is usually done in a lab, and then they're kept in a lab where we wait for them to grow bigger in order to outplant back into the wild. And what we found with microfragmenting is that when these reefs are cut really small, they actually grow a lot faster than a reef that hadn't been cut would. So a microfragmented coral might grow the same amount in 1 month that a larger coral would grow in 1 year. So that's why microfragmenting is being implemented in a lot of coral restoration efforts, because if we can help these corals grow faster, then we'll have more coral and we'll be able to outplant these corals and help restore the population faster. Another technique that's being used is supercorals. And basically what this is um, in labs, you're genetic engineering corals that maybe have higher heat tolerance or have higher abilities, like higher bleaching thresholds. They're able to withstand higher temperatures or they grow faster. And so these supercorals are genotypically better, and and if we can identify which genotypes are better, then we can outplant them into the ocean and hopefully they'll survive longer and thrive and do better. In fact, at UD, we're kind of using a mixture of these methods in order to help restoration efforts. And I'm not going to get too much into detail of it, but what we're doing at UD is we're actually studying the growth of microfragmented coral. So we have multiple individuals of coral, different genotypes, and we're tracking the growth rate of these corals over an 8-week period. And then we're studying their genes and their DNA, and we're looking for a gene called cyclin E. And basically, cyclin E is a gene that we think helps growth, helps increase growth rate. So what we were looking for is if the corals that we have that are growing faster also have a higher expression of cyclin E. And we have found that that seems to be the case right now. So cyclin E is correlated with higher growth rates. So what this would mean, for example, is that we could identify genotypes that have this higher expression of cyclin E, and we would outplant them into the wild so that way they would grow faster. Or we could stimulate the cyclin E gene in other corals to help them grow faster. So the goal here is really to just help identify which genotypes would be better to outplant into the wild to give them the best chance of surviving. So the second question I had to answer was question 8. And that is to describe the characteristics and ecosystem structure that defines polar marine environments and to discuss how global climate change and humans have influenced polar marine environments. So, the polar marine environment. The basic characteristics are that the marine environment has strong seasonality, meaning that from the winter months to the summer months, the ice coverage is vastly different. And so the ecosystem itself has a different structure throughout the year and alterations in their ice coverage. This in turn has a big effect on the planktonic population, which phytoplankton are extremely important in the food web to all organisms. And this has an effect on the phytoplankton because um, phytoplankton in the winter months live embedded in the ice. And then in the summer months, when the ice melts, these phytoplankton are released. So phytoplankton themselves have strong seasonality and because they have different assemblages from season to season. So this could have an impact on the food web. Another characteristic of the polar marine environment is that carnivores exert a very strong top-down effect. And a top-down effect basically just means that carnivores, because they're at the top of the food chain, can in turn affect levels below them on the food chain. And they could do this by, say, overfishing of these carnivores, like carnivorous fish. If there are less carnivores at the top of the food chain, then the bottom of the food chain could be affected, and the food web in turn could be altered. So next is the structure of the polar ecosystem. So the structure is that there's extremely cold seawater because it's in the Arctic. There's also a high level of productivity, and that's because, again, the thawing and freezing of the ice in the ocean, stores nutrients. So in the winter months, nutrients are stored in the ice. And then in the summer months, all those nutrients are released by the ice thawing, which puts a lot of nutrients into the water, which helps the lower level organisms like the plankton and the diatoms feed, which in turn helps support the rest of the ecosystem. So there's a high level of productivity in this environment. There's also a high level of organic deposition on the benthos, which is because when animals die in the polar environment, they sink to the bottom, and it doesn't matter if they're a really large whale or if they're a really small krill, they all sink to the bottom and they're deposited at the bottom. So that causes there to be a high level of organic deposition. But the worry with this is that without ice, without the freezing and thawing, there won't be enough nutrients in the water to support those lower level organisms. And in turn, there won't be, and in turn, the rest of the animal population will decrease, which will also cause the organic deposition on the benthos to decrease. And I drew this graph. So next we're going to talk about climate change. So first I copied this graph from Dr. Soper's lecture, and I just wanted to point out that climate change is occurring and it does have an effect on the polar environment. This graph shows the ice coverage on the y-axis over the years from it's 1978 to 2008. And basically this graph just shows that the ice coverage has decreased rapidly over those years, which is due to the warming temperatures. So this was actually documented by James Balog. James Balog is a photographer who made the documentary Chasing Ice. He also helped with the documentary Chasing Coral, which I used some of his information in the last question. But Chasing Ice was basically what he did is he used time-lapse cameras and he tracked the recession of ice in these polar environments over a few years period. And with his cameras, we can actually see specifically the decrease in ice in one area over many years. And the decline in ice is actually very, very rapid, and it's very eye-opening because of how much of a decrease we can see from his documentary. So we know that climate change is occurring, but what effect does it have on this polar environment? Because this polar environment is so dependent on the freezing and thawing of ice. Well, one thing that climate change causes is there's a big reorganization in the interactions between trophic levels. With climate change and less seasonality, we're going to see less interactions between these trophic levels because, for instance, diatoms, they're at the bottom of the food chain, and they rely on nutrients from the ice, the freezing and thawing. They rely on those nutrients to survive. So diatoms feed animals such as copepods or zooplankton, and zooplankton feeds fish such as herring, and herring feed those larger carnivorous animals such as seals. And so if we don't have this freezing and thawing, and there's a lack of nutrients in the ocean for these diatoms to eat, or these diatoms to get, and there are less diatoms, then there are going to be less of everything else in the trophic level. So the population of all these species is going to decrease. And the energy in each trophic level is also going to decrease. Another effect that that climate change could have on the polar environment is krill. Krill are extremely important in the food web because they feed on algae, but they also are consumed by many large predators. So because krill feed on phytoplankton, they rely on the freezing and thawing and the release of phytoplankton to survive. And if the freezing and thawing doesn't occur and there are less phytoplankton, then krill won't have a food source, and the krill population will decline. And we've actually seen that higher levels of ice actually equal higher levels of krill population. So it's expected that with these warming trends that we're seeing in the polar environments, there will be less krill, and in turn there will be less food sources for every other organism that relies on krill as a food source.